second summit. I think I can say from the point of view of everybody I've talked to that this meeting is going extremely well, and I think providing the framework for thinking about the use of gene editing into the future. Today's session will open with a panel of one person moderated by Dennis Lowe. Dennis.
So welcome to the first session today. So we have one distinguished panelist today. So it's Professor George Daly, who is the Dean of the Faculty of Medicine and the Caroline Hughes Walker Professor of Medicine at Harvard Medical School. So George is going to talk with us today about his thoughts on the current research and clinical applications, pathway to translation. So let's welcome Professor Daly.
Oh yes, how do I? Okay, wonderful. Thank you very much. So I'm giving a reprise on a session that I spoke in 2015, where I was asked to outline the plausible clinical indications, both for somatic gene editing, as well as the prospects for editing away heritable diseases at the embryonic stage. And I would say in 2015, that was a distinctly challenging task. There was widespread belief that the science was still inchoate and undefined, and many of the challenges were before us. And so many would have preferred not to even consider the prospect of editing of embryos. And then, of course, even in the case where you might accept that there was scientific feasibility, there's still dramatic ethical and moral debate about if we can do it, whether we should do it. And if we believe that there are indications that warrant its application, what are they? And how do we engage in a discussion that leads to the conclusion that there is societal consensus about the use of this powerful technology for eradicating disease at the embryonic state in a way that would be heritable? So I'm really going to be talking about what I think is the dramatic evolution that's occurred over the last 3 years. And I want to make some provocative statements. I think after yesterday, with the revelations of the births of gene-edited twins, I think the sort of tide was largely negative, and the prospect of anticipating ethical uses in the future really was almost set aside. But I think we do that with some risk. Just because the first steps into a new technology are missteps, it doesn't mean that we shouldn't step back, restart, and think about a plausible and responsible pathway for clinical translation. And that's what I'll try to articulate today. Now, going back into the statement from the organizing committee in 2015, we looked at the prospects for both basic and clinical research, and there was a strong sense that there needed to be further scientific investigation of the techniques, of the fidelity, measures of off-target effects, attempts to balance the risks and the benefits, not only for somatic applications, but also for the editing of human embryos. And of course, there was a strong call, especially given the sensitivity around any work with human embryos, there was a strong call for rigorous ethical oversight. And at that time, it was very clear that a prohibition was really called for. There was no sense that we had any deep understanding of the science, enough that would make it reasonable to move forward with establishing a pregnancy. And of course, in speaking about clinical use, there was wide expectation, and I will in fact briefly review the progress that has been made, but it was clear that genome editing for somatic indications was imminent, and that it should go forward under the existing regulatory frameworks that had been so successful in overseeing various gene therapy, and that at that time, given the risks, both known and unknown, the organizing committee made the very strong statement that it would be irresponsible to proceed with any clinical use of germline editing. Now, in the past 3 years, it's clear that now there are many, many clinical trials that use both the zinc finger nuclease, the TALENs, and more recently, the CRISPR. And I have to thank Matt Porteous, who actually gathered all of this data. And what you can see is that Sangamo has launched a number of efforts, interestingly, to target the CCR5 in somatic cells, with the goal of rendering cells resistant to HIV infection. And indeed, there has been a publication in the New England Journal of Medicine in 2014, which strongly suggested efficacy of this approach. More recently, there's been even more excitement, more enthusiasm, and I believe we'll hear later in sessions later this morning about the very exciting potential applications of gene editing for ameliorating sickle cell disease and beta thalassemia. Similarly, TALENs have been employed clinically. There's been some early data that has surfaced. And interestingly, at least 12 clinical trials of CRISPR-Cas9-based gene editing for a variety of different targets. Quite popular is the knockout of the PD-1, the sort of break, the checkpoint on the immune response that we know through antibody inhibition really stimulates a very potent anti-tumor response and is part of the real revolution in immuno-oncology. It may be that somatic gene editing will be the next step in that revolution. But interestingly, as noted by Matt Porteous, in doing this survey of these clinical trials, there's very little preclinical publication of the experience of these laboratories and the readiness for taking these projects forward. And to date, there's been no clinical publications reporting progress. So while there is clear clinical engagement, there is still a question as to how much transparency and access to peer-reviewed data. And I think that's an important message as we take these trials forward. Now, I think the lesson, though, and I think we're comfortable that the existing regulatory frameworks that have been in place, that have watched over the dramatic advances in gene therapy, primarily retroviral and lentiviral gene therapy, have worked and do provide the kind of oversight that we know accords patient protections and rigorous clinical analysis. But of course, the trade-offs with any regulatory system is it introduces a cumbersome, slow, and fairly conservative approach to this work. And we're constantly debating the trade-offs. The importance of regulation for ensuring patient safety and ensuring rigor, but sometimes coming at the compromise of innovation. Now, speaking then directly to the very, very strong statement of the organizing committee in 2015, where we used the word irresponsible. It would be irresponsible at that time, in 2015, to proceed with any clinical use of germline editing, unless and until, and there was a framework, there was a set of targeted goals that it was felt had to be addressed, and that included additional research so that we had a deeper insight into the safety and efficacy of the various gene editing strategies. There was an equally important, and I say still critical, role for further discussion, debate, and approach toward a societal consensus around which indications would be most appropriate. And then, of course, finally, and central to all investigation and all innovative medical therapy, is the importance of appropriate regulatory oversight. Now, at that time, 2015, December 2015, just 3 years ago, there was wide consensus that these criteria had not been met for any proposed clinical use of germline genome editing. And yet there was the call to realize that as the science evolved and as our understanding of the ethical issues evolved, that the clinical use should be revisited on a regular basis, and arguably this meeting is that chance to revisit. It was the call for this international forum that, in fact, led to this meeting. Now, in the last 3 years, it's quite remarkable how many different groups have weighed in on these concepts, and I'll highlight a few because I do think they speak to the rapidly evolving appreciation of the science and our now understanding, I believe, that we see gene editing not only as feasible, but as very potentially safe, certainly safe within the risk-benefit analysis for the treatment of severe diseases. So, in 2016, Jonathan Kimmelman and I anchored a study group of international scientists and ethicists assembled by the International Society for Stem Cell Research to look broadly at the conduct of both embryonic stem cell research and, more broadly, embryo research. It was clear to us that the debates about embryonic stem cell research really were founded upon the unique sensitivities of the human embryo. And that in whatever type of research was evolving, and embryo research was evolving far beyond the simple extraction of embryonic stem cells to present the actual opportunities for embryo manipulation, in particular by gene editing. So we broadened the call for embryonic stem cell research oversight to the more standard embryo research oversight, and really called for any type of research endeavor involving human embryos to be subject to this kind of rigorous and independent oversight. The ISSCR clearly called for additional laboratory-based research, including research into the modification of human embryos and human gametes, and it strongly stated that without further understanding of the risks and benefits, and without a further ethical consensus, that any attempt to modify the nuclear genome of human embryos should be prohibited at that time. Now, a little more than a year later, the study that had been called for and set in motion from the last summit was taken up by the United States National Academies, and it led to a similar endorsement of the importance of basic research, both for somatic and germline applications. And it similarly said that clinical trials for somatic editing should continue, subject to the existing regulatory frameworks. But interestingly, and I think the wording here was carefully chosen and is quite powerful because it suggests a strong step forward from the perspective of the last summit. And I read to you, given both the technical and societal concerns, the committee concludes there is a need for caution in any move toward germline editing, but that caution does not mean prohibition. It recommends that germline editing research trials might be permitted, but only after much more research to meet appropriate risk-benefit standards for authorizing clinical trials. This is a remarkably bold statement. We heard from many different countries presenting their regulatory framework yesterday that essentially prohibited clinical trials. And here we have a study which is saying the committee believes that the science is feasible with further analysis, and that it's time to consider the moral principles that would call into possible clinical use germline editing. And they set up a set of criteria. I won't belabor them. You can see them in the document. But they were criteria that adhered to universal principles: the absence of alternatives, the conduct of the research with strict transparency and independent oversight, and a number of others. But this set the stage for a possible translational pathway to germline editing. Further reinforcing, in 2018, just a few months ago, the Nuffield Council on Bioethics concluded as well, similarly, that we can indeed envisage circumstances in which heritable genome editing interventions should be permitted. So we have an evolution from might be permitted, meaning the possibility of ethical permissibility, to a call for almost an imperative for certain indications. That if we can solve the scientific challenges, it may be a moral imperative that it should be permitted. Now, the Nuffield report also highlighted the importance of the individual, the central concerns for the welfare of the individual whose genome would be edited, as well as societal concerns for social justice and social welfare, two important principles. So those themes of both scientific analysis and ethical consensus are critical to the evolution in these various opinions over the last 3 years. I point you to a very interesting review that was published in the CRISPR Journal that recounts and has a table detailing the 60-odd reports on human genome editing that have occurred over the last 3 years, and the vast majority of which indeed agree with the proposition that it is premature at this time, given the scientific uncertainties and the still lack of ethical consensus. It is premature at this time to proceed with clinical use. But there's a small number here in the blue that raised these same prospects, that should the science be deemed feasible, that the clinical use might be or should be permissible. So where has the science progressed? We've seen really quite remarkable presentations yesterday from some of the leaders in defining the technology. I think it's quite spectacular to see the array of different enzyme systems that have emerged, the notion of base editing. It's absolutely clear that this is a transformative scientific technology with the power for great medical use. There's also been quite a remarkable facility with being able to use this genome editing technology to engineer engineer whole organisms. Very, very soon after the first publications of the techniques, they were applied in mice, and then very soon thereafter in monkeys. And over the last few years, we've seen a number of very interesting applications to primates. And indeed, there is a deep investment in using genome editing technology to establish primate models of human disease. And I think that's going to be profoundly powerful for creating more predictive and more useful animal models. It's also providing us some of the technical feasibility for germline editing as applied to human embryos. The first reports, happening really just ahead of the last summit in 2015, were met with quite a bit of consternation, in part because the message was how unfaithful the actual editing was. There was a significant burden of off-target effects, and there was ongoing concern whether those issues could ever be resolved. And a number of groups have applied gene editing now to human embryo in the context of in vitro fertilization, attempting to determine variations on the protocol that would enhance the fidelity and reduce mosaicism. I think there's been an emerging consensus that the off-target problem is manageable, and in some cases even infinitesimal. And there's been some very interesting proofs of principle that there are diseases like thalassemia that could potentially be approached by this strategy. So in 2015, I laid out the prospects for how one could imagine assessing the safety of embryo interventions. Given the IVF process affords the prospect for embryo biopsy, either blastomere biopsy or trophectoderm biopsy, and the facility now with deriving embryonic stem cells, even from single cells, is high enough that one can, with great fidelity, be able to generate cell lines that allow you to do a significant amount of functional characterization, so that if one attempts genome editing at these early stages, and there are still variations on when the intervention occurs, and I would argue there's still much to be learned to optimize that. The main concerns after doing all of this analysis were really concerns for mosaicism. That is, if the gene editing approach was not 100% efficient at the sort of single genome stage, you might end up with a mosaic edited embryo, and because of sampling issues, you may or may not identify those cells that were effectively edited, and it could lead to some misleading results. So I think much of the focus has really now been around enhancing and optimizing the editing strategies at the earliest stages. And I think there's every prospect to imagine that even though one could not necessarily achieve 100% certainty for lack of mosaicism, it would get to the point where it could be very, very faithful. Now, of course, another strategy for which there has been considerable progress in the last several years is the ability to generate gametes in vitro. Those gametes can be derived from induced pluripotent stem cells, and those induced pluripotent stem cells can be subject to gene editing, and because they're clonable and immortal cells, they can be exhaustively characterized. And then, as has been shown by a number of groups, but most notably Mitanori Saito, these in vitro derived gametes have actually proven effective for birthing mice, and there are now reports of very similar biology applied to human iPS cells. So I think we're not that far removed from technical competency for in vitro gametogenesis, which is yet another vehicle for ensuring safe and effective genome editing. So I want to propose that it's time for us to at least consider a responsible pathway for clinical translation. And what would such a pathway entail? I would say that we are still, as of this second International Summit on Human Genome Editing, lacking a deep scientific consensus for the optimal methodology for editing human embryos. There are still concerns for mosaicism. There has been relatively few publications that have exhaustively analyzed different strategies. I would say we still lack a scientific consensus on the optimal methods for assessing the fidelity of gene editing. There are many different techniques, but as we spoke about yesterday, evolving towards a standard that could be applicable in a clinical trials still required. And then I think equally important is standards for assessing the technical competence of the practitioners that would be given the right to go forth with a human clinical trial. In the prior analyses of embryonic stem cell ethics, it was strongly stated by the ISSCR that one had to ensure that the individuals that were given the right to derive embryonic stem cells and to use the uniquely sensitive materials of human embryos, they had to be expert. They had to be trained, and this is in part something I think that would be required for any clinical indication. So I want to suggest that I do think it's time to move forward from the prospects of ethical permissibility to start outlining what an actual pathway for clinical translation would look like. What would be the regulatory standards that a group would be held to in order to bring this technology forward? And equally important, in fact, maybe even more important, if we are ever going to practice this with a deep sense of social justice, there needs to be additional definition of the distinctions between the permissible and impermissible. And so I return to a slide that I showed in 2015, which acknowledges the fact that there is a range of potential indications, some of which will curry much greater enthusiasm than others. It would surprise me if the first indications that were thought the most feasible were not those indications that represented very devastating and life-threatening diseases. And under situations where the alternatives were not optimal, and even though there are alternatives such as preimplantation genetic diagnosis and embryo selection for autosomal recessive diseases, we all know that that methodology is itself fraught with inefficiencies. And it may be that coupling IVF to gene editing may one day be the more efficient and more effective way to eradicate these diseases. And of course, there's ongoing development of somatic therapies. And there are vanishingly rare, but still existent, and rarity alone is not an excuse to not pursue a medical treatment. But there are vanishingly rare couples affected by genetic disease, perhaps where both are affected and where the only option is genome editing. And then, of course, somewhere in the middle are the modifying disease alleles. Interestingly, CCR5 being one of them. These are not themselves 100% predictive of a disease condition, but rather disease modifying. And there have even been discussions that we as a species need to maintain the flexibility in the face of future threats to take the control of our own heredity. And then, of course, I think still not in any way a plausible approach would be to entertain the use of this powerful technology for enhancements. And so I do believe that we're at the time where, if we're going to imagine a pathway towards clinical translation for germline editing, solving and addressing these deep issues are essential. And I want to leave my last slide with an important message and a cautionary tale. The fact that it's possible that the first instance of human germline editing came forward as a misstep should in no way, I think, leave us to stick our heads in the sand and not consider the very, very positive aspects that could come forth by a more responsible pathway towards clinical translation. And since this conference has occurred really as part of the intellectual tradition dating back to the Asilomar Conference in 1975. I think we as a scientific community embrace the long traditions of self-regulation. It is one of our professional norms. But what this requires is it requires transparency. It requires each of us in the scientific and clinical community to invite external scrutiny, peer review, and oversight. And it is against these standards that I think are widely accepted in the international scientific and bioethical community. It is against these standards that we judge the current revelation of the first clinical use of germline editing. And I say that I believe we as a community can do better. So thank you very much, and I look forward to taking questions.
So please, so now we'll start the panel discussion.
I reserve the right to call on people in the audience.
So are you worried that with this first misstep, some authorities might actually just have a reflex action and suddenly institute some very tough rules, which is going to set back the field for a few years?
Yes. Yes, I do think that is a concern, and it is part of the risk to society that the oversight process should actually consider when doing an evaluation of such an innovative clinical protocol. Because you're not only assessing the risk to the individual, but you have to actually think about how a negative outcome could impact the future of a technology. It's a very compelling and interesting concept, but if there's a technology that has the potential for widespread good, for the relief of disease, relief of suffering, it's terribly unfortunate if an initial misstep leads to an overall suppression of interest in that technology. And so, yes, I think there was a strong sense yesterday that the current standard is impermissibility. But once again, I hope we don't just stick our head in the sand.
Okay, thank you. So we'll start one question on that side first.
Can you hear me? Okay.
You can say it and I'll repeat it. No technology is flawless.
All right, thank you. Tamara Lysart from the National University of Singapore. Thank you, George, for a really interesting and enlightening and forward-thinking overview of the clinical applications, especially in light of the events of the last 48 hours. I just have one comment, first in relation to the number of kind of reports, bioethics reports that are kind of coming out and indicating that there may be some way forward in terms of germline editing. While there may be some consensus building up in sort of bioethics circles, I think that the media response we've seen in the last day or so strongly indicates that we are far, far away from a social consensus. So that was just a word of warning about that. My question relates to your comments about responsibility and what that is going to mean for the scientific community. Of course, if anyone saw my talk yesterday, I'm very interested in this concept about responsibility, and so not just as being a nice rhetorical word we can tack on to things like innovation and research, but what does that actually mean in terms of how we practice good science So in addition to the technical standards and research that you mentioned in your talk, could you reflect on what it will mean for scientists to practice social and moral responsibility in this area? Thanks, George.
Yeah, it's an excellent question. So in the point of social consensus, I think it is true. I don't believe there is yet social consensus on either the permissibility of any indication or any specific indication. So it's another reason for ongoing discussions like the one we have here. And then on the issue of responsibility, it's absolutely clear, and yet there's a tension. Scientists are by nature intrepid investigators. There's a reward to being a pioneer. There's a reward to being first. But I think we as a community have to accept the responsibility, especially in the area where there's an intervention that may generate a certain degree of controversy, that we be willing to be subject to external oversight. I think it's a core principle to be given. We could argue it this way: to be given the privilege of doing science, often at the expense of the public. To be given that privilege, we have to cede the right for independent oversight. And I think that scientists who go rogue, and I know we're going to have a session where we'll be talking about the sort of ethics of rogue science, it carries a deep cost to the scientific community.
Thank you. So we'll take another second question on this side.
That one's not working either.
Not working? Please. You can use mine as well.
I think the reporters outside have taken all of the microphones.
Thank you for the excellent
more reporters than
presentation. My
microphones.
question is to George as well. So when you mentioned the clinical application should be permitted. So my question is, if we should distinguish to the between the research and therapeutical therapeutical purpose, because for therapeutical purpose, we should focus on the solve the severe disease, genetic disease. But for research purpose, is focused on obtaining generalizable knowledge. So if we should distinguish this, and if we should have the different principle, ethical principle, or the rules about that, and how to use it. Thank you.
I think that point's very well taken. The difference between the motivation for general knowledge versus the actual prevention or relief of suffering as in the treatment of disease. They're two different goals.
Hi, Sarah Chan from the University of Edinburgh. Thank you very much, George, for that really enlightening talk. I wanted to just raise a couple of doubts about your suggestion that in vitro gametogenesis and the generation of embryos from in vitro gametes might provide a way forward for ensuring safety and accuracy in producing germline genome-edited children. In theory, it sounds very sensible, but thinking about what's a realistic translational pathway from this point in time forward, I am really uncertain that IV gametes are going to provide that. And the reason is that we, I think, technically and ethically are much, much further away from being able to create babies from embryos, from in vitro gametes, than we are from just going ahead and applying the gene editing technology we now have to embryos produced in the conventional way. I cannot imagine what the convincing reason for the first in-human trials of in vitro gametogenesis followed by IVF would be. I wonder if you would like to comment a bit more on that.
Well, infertility is a clear indication that would be pursued. But I agree. I mean, I think that there's dual strategies of enhancing the fidelity of editing at the pronuclear or zygotic stage to ensure uniformity of gene editing versus doing it through an in vitro derived gamete. But I agree that the science of in vitro gametogenesis is somewhat more futuristic, but the progress there has been truly startling, truly remarkable, and I think it's going to be driven by the interest in treating infertility.
Thank you.
Hi, Rachel Harwitz. I'm the CEO of Caribou Bio, a CRISPR company in Berkeley, California. I think inherent in the thinking behind how you put together a translational program is the assumption that parents have the affirmative right to a biological child. I was wondering if you could talk a little bit about that, given that, of course, there are other approaches to building families, adoption, and other strategies.
Yeah, I can't say that I'm an expert on that concept, but there are individuals who are, and maybe some of them are here. But I can say, speaking as a parent, there is a strong desire and drive that does create, at some level, an imperative.
Thanks.
Now, since we have started about 10 minutes late, so maybe we just press on for that 4 questions, and then we finish the session after that. Okay, so please.
Okay.
Yep.
Thank you so much. I'm Han Zhiyu from Zhejiang University. I'm a research fellow of public policy. So my question is, I really appreciate you mentioning about the self-regulation and the consensus for the scientific community on this issue. But my question is whether you think it could be a risk if someone can just ignore all the norms and standards in our scientific community because of the market, the capital support, industrial support. If yes, so what could be the response, or what things can do, like for the academic community that can respond to this kind of like industry or market response? Because I just know that after you mentioned like the mid-step of the event 2 days ago, that there in fact there is a very positive response from the market, from the capital market. So I want to know what is your opinion. Thank you.
If I understand, you're asking about how does one balance the call for ethical oversight and a responsible way forward with the kind of market-driven forces and the possibility that industry could move forward. I noted in passing that of the CRISPR trials, there wasn't a lot of publication associated with those trials, and I think there is a need for more transparency in general for commercialization and commercial types of trials, but especially for the first uses of highly innovative technology, and a technology, if we were to apply it to the germline and to heritable editing through embryos, that I think raises the bar for having transparency and willingness to subject to external oversight. And maybe for that reason, it's best done within an academic realm rather than an industrial or commercial realm.
Okay, thank you.
And that will be debated.
Thank you.
Thank you. The next panel, please.
My name's Evan Kirksey. I'm an anthropologist from Deakin University in Australia. And I just have a question about your optimism about the possibility of universal principles and consensus. So Immanuel Kant is one philosopher who's famous for, trying to come up with universal ethics. I think some contemporary philosophers, namely Isabel Stengers, are maybe more realistic about how contested values persist. Looking around us and at the panel tomorrow, or the panel yesterday, we saw all sorts of values and moral sensibilities from Islam, from Christianity, from secular scientific ethics, from Neo-Confucian ethics, in an era of the China Dream that aims to disrupt contemporary Western modernity with dreams of an Asian futurity. So I'm just questioning your optimism about consensus.
It's funny, it's unusual to question someone's optimism, I think. I'm glad that I can still be optimistic. You talk about, the traditional tensions in sort of Benthian utilitarianism and Kantian deontologic approaches. I think we aspire to universal principles. I think we aspire to the Kantian, but in everyday life, especially as a physician, I use sort of utilitarian principles all the time. But I think that's part of what was so exciting about some of the presentations yesterday, as we thought about many different international principles, and we started to think about the different religious principles and the different philosophical principles. It's part of the reason why we have to bring together communities like this to struggle with those issues, because we won't get to the point where there will be acceptance of a technology if we can't find some common ground. And unfortunately, we've had other potentially very powerful technologies that had the force of good. You know, genetically modified organisms, in its highest application, could, you know, feed starving millions. And yet it's been very, very hard because of missteps early on in their introduction. There wasn't broad societal consensus for those introductions, and that's been terribly it's been painful for the world community. I hope we don't go that way in the application of gene editing.
Thank you. Next gentleman.
Ben Hurlbut, Arizona State University. My question follows up very directly on that question and this answer. And my question is, in effect, what you mean by consensus. Consensus about what and among whom? You talked quite a lot about scientific consensus. Professor Baltimore, who's sitting in the front row here, I think will remember that in 1975, getting an unruly group of about 100 scientists to arrive at a kind of univocal consensus took quite a lot of work and a certain amount of, I won't say coercion, but maybe persuasion is a better word. And so, of course, there are asymmetries even within a vanishingly small scientific community. The community now is much, much larger and much, much more heterogeneous. And so I wonder just within the domain of scientific practice about the particular issues that you mentioned, what consensus looks like, and in effect, who sets the agenda for asking the questions that produce a consensus. And so then the second part of the question relates to the quote, the issue of quote, broad societal consensus. Do you conceive of that as following after the issues of scientific consensus? In other words, matters of scientific consensus are achieved, and then the question of broad societal consensus is in effect a question about consent. Do we allow these now perfected techniques to be employed? Or do you see that question of societal consensus as informing the very questions that get asked in scientific arenas about what we need to know, how we should know it, and to what end?
Right. Leave it to a philosopher to pose that question. You're pointing out the obvious, which is that consensus is virtually impossible, but it's still a guidepost. It's an aspiration that I think we should seek. Scientific consensus has to be sought contemporaneously with the moral and ethical consensus. There has to be at least an attempt to come to some broader understanding. It may not be universal, but it could be broad about the scientific principles, the sort of nature of standards. We do this all the time in lots of different interventions, whether it's in vitro fertilization or whether it's various treatments for a range of diseases. You might not approach consensus, but you will continue to strive for the kind of broad acceptance that will lead to greater embrace by the community, especially for areas like this. I think we've seen that evolution in vitro fertilization. It's not clear there was consensus when it was first rolled out, but I think there's widespread acceptance today. I think we can do it better the next time, and that's why we aspire to have meetings like this. And our last question?
Yes, good to hear your comments, Dr. Daly. I'm J.D. Hansen from the International Center for Technology Assessment. I'm one of the co-authors of the Principles for the Oversight of Synthetic Biology, which is signed on to by 120 civil society groups around the world. First principle is the precautionary principle. The second principle is binding regulation. What I'm hearing from you is an incredibly tepid response to the first major failure of the self-regulation principle that this community has been hyping for a long time. One of the challenges we've had with genetically engineered plants and animals is that we have a scientific and industry group that has been imposing regulation. What I hear you saying is that despite this major failure, the US people, the National Academy people here have not denounced this, but said it's a little problem. The National Academy of Sciences report on gene editing for conservation purposes is much more pushing for strong regulation than I hear you today. Why don't we need strong regulation now? Why don't we need a moratorium until we get it?
Mm-hmm. So I don't think a single practitioner who goes against the sort of the norms of a field represents a gross failure of self-regulation. I think we aspire to self-regulation in as many instances as possible. And the fact is, one practitioner is unlikely to have as profound effect as the entire community. And so if the entire community, or at least the majority of the community, buys into common principles of regulation, as the gene therapy community did early on, I think you have approached as closely as possible to an ideal. At the time where there is widespread flouting of these common principles, then I think you do need binding regulation. And we've seen that with the proliferation of unproven stem cell interventions. I've lived through that. We attempted to define broad principles that would guide clinical translation of these new therapies. But what happened instead in many instances was this rise of this sort of unregulated economy. But thankfully, Gottlieb and the US FDA has actually started to speak out very aggressively in favor of binding regulation or enforcement of that binding regulation. So these are complex issues, but I do think the fundamental principle of self-regulation is something the scientific community has adhered to, and I would continue to defend.
So thank you very much. So with that, we'll draw this session to a close. So let's thank Professor Daly in our usual way.